So this video lesson is on the last section of chapter 1, which deals with the cell structures. So we'll be looking at basically what most cells have in them, and then we'll talk a little bit about some differences between plant and animal cells, and then I'll probably do a separate little video lesson for the prokaryotic cells. So this one will focus mainly on eukaryotic cell parts, but let's go. So the essential tasks for living things, and so therefore for their cells, are to obtain food and energy, to convert that energy from an external source to a usable form. So for instance, when we take in food and we break it down, we are making it go from a certain chemical form of energy in the bonds of food to the bonds in ATP, which is our special energy molecule that we can use really quickly in our cells in order for us to have the energy to do work or to get the jobs done. Another task is to build and maintain molecules that make up cell structures. So from what we take in our diet and from what we build within our cells, then we can make other molecules that are responsible for anything from maintaining the cell structure itself to carrying out certain jobs that are in the cell. They carry out chemical reactions. They have to eliminate wastes so they don't build up, and they reproduce. So the cells must reproduce in order to allow for repair and replacement and basically to keep the species going on. And they need to keep records of how to build structures. So they tend to do that through molecular way. So they keep information in their DNA so that the next cells will be able to keep building those structures, and every cell would have those instructions if they need to build certain things. So there are two basic cell types: prokaryotic cells and eukaryotic cells. So to understand the difference, it's important you understand what those terms mean. Karyote refers to nucleus, and pro means before. So prokaryotic cells, we think before the nucleus, they have no nucleus. So that's the major difference between the two. Eukaryotic cells, eu means true. So they have a true nucleus inside of them. Along with that, they have other differences. So prokaryotic cells are smaller than eukaryotic cells. They have a much more simple structure. They don't have all the different organelles that a eukaryotic cell would have. They have something in common would be that they have ribosomes, but they don't have membrane-bound organelles like the complex eukaryotic cells do. An example of a prokaryotic cell would be bacteria. Eukaryotic, the cells of plants, animals, fungi, protists, all the 4 kingdoms that are eukaryotic. Okay, so we're going to look at more specifically the cell structures. And all cells, whether they are eukaryotic or prokaryotic, have 3 parts: a cell membrane, cytoplasm. Cyto means cell, so the living tissue within the cell, and cytoskeleton. So if you think of what our skeleton does, it's similar, but on a much tinier scale inside of the cell. Okay, so it gives it basically structure and shape and support. So we're going to focus mainly in this video on eukaryotic cells, and then I'll do a separate little video depending on time, I'll do a separate little video on the prokaryotic cells. So in a eukaryotic cell, remember it means true nucleus, so they have a nucleus. So we'll go and take a look at the nucleus. And the nucleus, if you remember from learning about it in junior high, the nucleus is usually pretty easy to pick out. It's usually a darker, larger organelle inside of the cell. Okay, it's nicknamed the control center because it is the site where you'd find the chromosomes or the DNA. Basically, without it, the cell would die. Two major functions include storing hereditary material, or the DNA, and so because it has the DNA, it also is important because it coordinates the activities in the cell. So that can be anything from growth to metabolism to synthesis of proteins to reproduction. There's just another image showing a nucleus. Inside of the nucleus, you find chromosomes. The chromosomes are the hereditary material. They are found in the nucleus of all living cells. And basically, they're made up of DNA wound around proteins. So here's the DNA, shown in the green, and they're wound around these histone proteins to give a more complex, coiled up structure. So we can only see them looking like these X kind of shapes whenever they're supercoiled during cell division. The rest of the time, they're you can't really see them. We wouldn't be able to see them in a cell, in the nucleus, because they're more, not as condensed. They're thread-like. Okay? DNA contains the genes that we talk about that provide instructions to direct all cell activities. And the way that they do that, we talk about more so in the grade 12 biology program. But it's pretty neat, pretty amazing that really 4 different nucleotides that make up DNA. It's a very simple structure, but it's very strong, I guess you'd say, in terms of molecules. They're hard to break down and hard to make changes to, but those things can happen. So it's important because we want it to be maintained, and with some adaptations, we want it to be able to be passed on and allow our offspring to be kind of like us, or at least have all the properties they should have to live a good life. So, also inside the nucleus, we have the nucleolus, and the nucleolus is the nucleus blown up, and then the nucleolus is a dark body inside of the nucleus. You don't always see the nucleolus, depending on the quality of your microscope. Okay, so here's the nucleus and a cross section of it, and inside of it would be the nucleolus. The nucleolus is basically it's involved in ribosome assembly. So it helps to make the parts of the ribosome, but it's also involved in putting the ribosomes together so that they can go out and do their job. So now we're going to take a look at some of the other organelles. So that was the nucleus and the things you find in the nucleus. We're going to take a look at some of the membrane-bound organelles and also some of the non-membrane-bound organelles. So that just means whether or not they have a membrane around them. So we'll take a look at the mitochondrion first. And if you remember from junior high, the mitochondrion is nicknamed the power plant of the cell because it's the organelle that undergoes a process that allows us to have energy to do whatever we need to do. So if it's a muscle cell where they're working all the time in order for us to move and run and do lots of things, then it would have a lot more mitochondria than, say, a skin cell, because it's not undergoing quite as much activity as a muscle cell. So the job of the mitochondrion is to supply the cell with energy, and it does that through a process called cell respiration. So that is an important term, cellular respiration. We're going to learn more about that in chapter 2-3. Again, like I just said, the number of mitochondria in a cell depends on how active it is. So there'd be lots in a sperm cell because they have a long journey. Lots in muscle cells and liver cells, not so much in, say, a skin cell. So this is what it kind of looks like. It's oval shaped, and inside of it, it has two membranes. And the inside one is kind of all folded up, and that's important because it gives lots of surface area so that we can have more and more reactions to give us more and more energy. Okay, the endoplasmic reticulum, the ER. Okay, if you're looking for it in a cell, it looks kind of like a ribbon, big ribbon, and usually lies fairly close to the nucleus. Its job in general is to transport substances made around the cell. So there's smooth ER and rough ER, and basically the difference is what's associated with it. So rough ER, this is rough ER. Up here you can see it a little better. So it has ribosomes that are attached to it. And ribosomes, their job is to make proteins. So rough ER then, since it has the ribosomes attached to it, it will transport proteins. The smooth ER, on the other hand, has enzymes that make fats, phospholipids, and steroids, so lipid molecules, and it transports the fats through the cell. Okay, so rough is responsible for the ribosomes make the proteins, and they put them into the rough endoplasmic reticulum so that it can be transported through the cell. Smooth makes lipids and adds things to them and transports these lipids or fats through the cell. So we said the endoplasmic reticulum looks like kind of like ribbon candy, like ribbons. But if you look at, let's see if I can make this a little bigger. This structure here, and we'll look at it in some other cells too. This structure right here, it looks like, there. It looks kind of like a stack of pancakes. And there's usually vesicles or little circles that are coming off the ends. They pinch off of the ends because the job of the Golgi apparatus is to package things and send them where they need to go. Okay, so it sorts things and packages them and sends them to where they need to go. So it's kind of like, I liken it to a post office. So they sort the mail and they package them into sacks and then they send them to where they need to go. Okay, so it'll receive things from the endoplasmic reticulum, so some substances that need to be sent other places will go from the endoplasmic reticulum to the Golgi apparatus and then be sorted and sent other places. Sometimes it's even called the Golgi body, and the reason it's called Golgi is because the fellow that identified it, his last name was Golgi, and that's a lot of the times where the names come from. The lysosomes are nicknamed the suicide sacs or garbage collectors of the cell. They're basically small, round bodies. They're made from, well, their contents are made in the endoplasmic reticulum, and so they're formed they're formed and break off as transport vesicles, and then they go to the Golgi apparatus. And then when the vesicle leaves the Golgi apparatus with the enzymes in it, it's referred to as a lysosome. So the lysosome has these enzymes in it, and the enzymes are kind of like, if you think of Pac-Man, then their job is to gobble up and break things down. So the role of the lysosomes is to fuse with other cells, like here's a food particle that's come in, a food vesicle. So inside of this vesicle there's food. So if the lysosome fuses with it, then the enzymes that were in the lysosome will act like Pac-Man, and they'll break down the food particles. So that's one job. Another job is they will go and join up with old organelles or damaged parts, and they'll break them down so that the parts can be recycled. They also will join up with a white blood cell that has engulfed a bacteria, for instance, and so they'll break down the bacteria or whatever the white blood cell has we'll say caught. So here's a little animation. It'll make it a little easier to visualize. Oh, there's the sound. Hold on. Need sound. Let's try that again.
Lysosomes are membrane-bound vesicles that contain hydrolytic enzymes. These enzymes digest particles or cells taken into the cell by phagocytosis. They also digest old organelles such as mitochondria. The hydrolytic enzymes that degrade proteins, nucleic acids, lipids, and carbohydrates are formed in the endoplasmic reticulum and then transported to the Golgi apparatus by transport vesicles. The lysosomes arise from the Golgi apparatus. When particles such as viruses or bacteria are ingested by phagocytosis, the lysosome fuses with the particle-containing vesicle, called a phagosome, and delivers the hydrolytic enzymes. Lysosomes also fuse with organelles such as defective or worn-out mitochondria. This results in the destruction and recycling of these structures.
Okay. So that is the lysosome. Oh, and the reason they're often called suicide sacs is that if they were to open up and release their enzymes inside of the cell, then they can break down their own cell parts. So they can basically, they're important when a cell has to be broken down, or when a cell has gotten kind of too old, then the lysosomes will actually break down all the cell parts and recycle. And if they get released by accident, then they would be able to destroy the cells by accident. Oops. Where did my link go? There used to be a link on here to go back to the page. I'll have to find it. Okay, so that was the lysosome. Lysis means to break apart. So a lysosome's job is to break things apart. Because of the enzymes that are in them, then they can break things down. A peroxisome, on the other hand, they can look similar to a lysosome. They're small, round structure within a cell, and they help to get rid of toxins. That's their job. When a toxin is taken in, it turns the toxin into hydrogen peroxide. So that's where the name peroxisome comes from. The peroxisomes then break down the hydrogen peroxide and turn it into water and oxygen. So these are very common in the liver and the brain and kidney cells. the liver is our really important organ for detoxifying your body. So it's important that they have lots of these there so that they can break down the toxins and make things safer to be transported or excreted from our bodies. Okay, so we're going to look at a vacuole. Now, sometimes lysosomes, peroxisomes, vacuoles, and vesicles are hard to tell between on cell diagrams. So, in class, we'll probably look at a number of them and talk about how you can tell some differences between them. And some of them you really can't unless I put some kind of identifying things in, or I might even accept one or the other answer, depending on what you say. So, a vacuole is basically a membrane-bound storage vesicle. So it is a vesicle, but it's for storing things. There's other kinds of vesicles that are for transport, but these ones are storage vesicles. So they might store food or water or wastes. In animals, they tend to be more numerous, but very small. In plants, they tend to be large. So they have a large central vacuole, and usually it's important for storing water, but also, if you look at it, it takes up a bulk of the space inside of the cell. And if it's filled with water, then that kind of pushes the vacuole, but also the other cell parts against the cell wall, and that's what gives the plant what's called turgor pressure, or the pressure to be able to stand up and be healthy looking. If they don't have enough water, or water's left for some reason, maybe there's too much fertilizer, then there's less water in there to create the turgor pressure, and so the plant will tend to wilt. So that's why it's important to make sure you water your plants enough, but not too much. So the vacuole in the plant cell tends to be large. It's usually for storing water, but sometimes it'll also store wastes like crystals that the cell makes and other toxic wastes. And vesicles, we usually say, are membrane-bound organelles that transport materials through the cell. So they can be transport vesicles or storage vesicles, which are called vacuoles. So we tend to differentiate between them that way. If it's for storage, then we call it a vacuole. If it's for just for transport, then we tend to keep calling it a vesicle. So vesicles, transport vesicles, are moved along microtubules. So if you think of a microtubule like a highway that they can travel on, then the vesicles are kind of pulled along these highways, these tubules, by little motor kinesins. They're proteins that their job is to basically move things along. So there's different types, like Golgi vesicles, and there's the Golgi apparatus, and there's the endoplasmic reticulum. So the Golgi apparatus, there's vesicles that are coming off of the Golgi apparatus, and they tend to take things out to the cell membrane. There's transport vesicles, which are formed by the ER, and they transport materials through the cell, often to the Golgi apparatus or other parts. There's pinocytotic vesicles and phagocytotic vesicles, which are basically formed during the cell pinching materials inward, or that's in the case of pinocytosis, or phagocytotic, where the cell kind of moves out around things that are bigger and pulls them in. Okay, so this is showing formation of a pinocytotic vesicle. So it pinches in some more, it's almost pinched off, and there it is inside of the cell. And I thought I had, here, I'll just go out and show you. We'll show you a little animation of it. Okay, so this is the kinesin, and this is the transport vesicle, and this along here would be the microtubule. So it's like the highway, and this little guy is almost like they walk them along, but it guides the vesicle along the microtubule highway as it's going through the cell. And we're going to watch a video later on where you can really see them. It's kind of cool. So that covers all the membrane-bound organelles that we're going to talk about right now. In terms of non-membrane-bound organelles, so these ones don't have a membrane around them. Cells have ribosomes, which are often nicknamed the protein factories because their job is to make proteins, so protein synthesis. So they're in making proteins, they translate the code that comes ultimately from our DNA. So our DNA instructions are carried out through the building of proteins. Okay, so we talk in a lot more detail about that in the grade 12 program, but it's pretty neat. Pretty neat to learn about. Ribosomes are made up of two parts. There's a big subunit, a 60S, and a small subunit, a 40S. One part decodes the genetic information, and the other part kind of adds the building blocks of the protein called amino acids. It adds them to the chain. Where can they be found? There's some that are found attached to the endoplasmic reticulum, so it makes it look bumpy, or we call it rough. And they make protein that is destined for inside and outside the cell. But there's also cytoplasmic ribosomes, which just means that they're floating around in the cytoplasm, and they're like little dots all around inside of the cell. And they make protein just for inside the cell. We also have cilia, and cilia are basically short hair-like projections that work like oars. So they're usually plentiful, many of them. They're found in animals, but rarely in plants. They're composed of microtubules, and basically they work by beating in coordinated waves. So they might be used for movement of some organisms, but they might be a way of moving stuff along or moving them in a certain direction. And there's also some associated with sensory organelles. So some examples, this little guy called a paramecium, he is covered. You can't really see them very well because they're very tiny, but he's covered. He's got cilia all around the outside of him, and they beat to help him move. And they also will use them, this is called his oral groove, so it kind of brings, by beating the cilia, it's not a he, but I'm just saying, anyway, by beating its cilia, it causes a current and kind of brings food down into this groove, and at the end, it's kind of like a mouth, but not really a mouth. We also have cilia that line our respiratory tract, so inside of our nose all the way down to our lungs, and so they help to filter mucus and dirt. And we have cilia along our oviducts, for instance, that help to move the egg or the ovum down along the oviducts from the ovary to the uterus. So there's lots of different places where you'd find cilia. Flagella, on the other hand, flagella are usually long and usually only have maybe 2 to 5 of them, depending on the organism or what kind of organism has those particular organelles. So They're really long in comparison to cilia, and they work more whip-like in their action. So they are whip-like, and they tend to propel the cells forward. They're also composed of microtubules, so both cilia and flagella are made of microtubules. And here's some examples. So these are all different types of sperm. This is showing mammal sperm. I can't remember which kind, if it's human or if it's dog. But basically, this is showing the building of the flagellum. And these are the microtubules, and they're arranged in such a way that they form this long structure for moving. Okay, so this is just a diagram showing the difference. This is a flagellum on a euglena, and this one has tiny, tiny little cilia all around the outside. And the type of movement is more like oars, the way that they work, whereas the flagella, it's like a whip, and it whips around, makes them move. And microtubules we'll be talking about in a little bit, but microtubules are basically a type of protein. And for some reason, my links are gone from the page 2. So we've talked about now all the membrane-bound and non-membrane-bound organelles that are common to eukaryotic cells. Now we're going to talk about ones that are unique to animals or plant cells. So unique to animal cells, centrioles. Centrioles are also made of microtubules. They're short, cylindrical structures. So if I take this away, you can see there's 9 sets of 3 microtubules, the way that they're arranged. Okay, 9 sets of 3, or 9 triplets, as it says in the description. And they're found at right angles, or they're usually found in a pair at right angles to each other inside of the centrosome. And the centrosome, or microtubule organizing centers, are important in cell division. Okay, so if you remember from talking about mitosis and meiosis in junior high, the centrioles will migrate to opposite poles, and they're important in formation and direction of the microtubules in the spindle. So they are important for organizing the microtubules that kind of pull the chromosomes apart. Plant cells have a number of unique features. So they have a cell wall. Okay, this is the main difference between plant and animal cells. The cell wall in plants is composed of cellulose, and cellulose is important for us in that it provides roughage. We can't break it down. We don't have the enzymes to break it down. Excuse me. So when we say need to get some fiber, then that's basically we're eating lots of fruits and vegetables because they're plants, and they provide us with cellulose, which we can't really break down, but it's really important in that it provides roughage, or the fiber, to kind of help flush through our digestive tract. Kind of keeps cleaning your pipes, as they would say. Okay, so there's 2 layers to the cell wall, and the function of the cell wall is basically to provide skeletal support, in other words, structural support, and it helps to maintain a fixed shape. It offers protection to underlying cell parts, and it prevents the cell from bursting due to osmotic pressure. Like if that central vacuole kept filling with water, then the water and the pressure against the cell membrane alone would cause it to burst, and the cell would just no longer exist. It permits the turgor pressure, which we talked about, and that helps to keep the plant rigid. Another unique feature in plants is organelles called plastids. And if you notice, plastids, when you look at specific plastids, they all end in plast. Oh, wrong connection. Oh, my links are messed up. So, of the plastids, there's 3 types we're going to look at: chloroplasts, amyloplasts, and chromoplasts. So, chloroplasts, you've probably heard of before. Chloroplasts are basically the organelle that's responsible for photosynthesis. So they're not found in any animals, but they're found in the green parts of plants. Photosynthesis is basically the process where we get light energy from the sun, not we, but plants and other organisms that can undergo it. Will take light energy from the sun, and they'll convert it to the chemical energy in glucose or food molecules. The chloroplasts contain a pigment called chlorophyll, and the chlorophyll is really important for trapping the sunlight, but it also is the reason why that part of the plant that has them appears green. So it reflects the green color that we see. Inside of the chloroplast, so here's a chloroplast in a plant cell. Inside of the chloroplast has a double membrane, like the mitochondria does, but inside of it are stacks of thylakoid discs, or thylakoid membranes, and each stack is called a granum. So all the stacks together, plural, would be grana. And the reason they have those is, again, to increase surface area, so there's lots of surface area there to trap the sunlight and undergo the reactions to create glucose or foodstuff. Amyloplast, so the chloroplasts we see as green. Amyloplasts are a type of leucoplasts, and leucoplasts are colorless plastids. So they're found in the cytoplasm of some plant cells, depending on their role or where they're found in the plant. Their job is to transform glucose into starch, and they produce and store that starch. So you would find a lot of amyloplasts in a potato cell. So we're going to be looking at potato cells, and this is kind of what it looks like. They look very clear overall, and then inside of them you see all these different kind of shaped, oval-shaped things, and those are all amyloplasts, and they'd be full of starch. Chromoplasts, chromo means color, so chromoplasts are plastids that are found in photosynthetic organisms, and they're responsible for synthesis and storage of pigments. They give color to the plant parts. So technically, a chloroplast is a type of chromoplast, but most of the chromoplasts that we call chromoplasts give the other colors. So they're found in colored organs like the fruit and petals. An interesting fact, when we have ripening or going from green to color, it's a conversion of chloroplasts to chromoplasts. So they break down the, why am I saying we for everything today? Anyway, they break down the chloroplasts, and so then the other chromoplasts are reflecting other colors like orange and red and yellow and other colors like that. The last of the structures that are unique to plants we're going to talk about are crystals that they form. So they're druce and rhaphiid crystals or rhaphite crystals that are basically calcium oxalate crystals that are waste that some of the plants will produce, and they are stored in the large central vacuole. And they sometimes they're just produced as a byproduct and happen to have this property, but they will often deter other organisms from eating them due to bitter taste. And sometimes they're actually toxic to the organism that's trying to eat them. The raphites are needle-shaped, and they're kind of sharp. So if something tries to eat them, then they're going to kind of sting them or hurt them as they're trying to swallow them. Druce crystals can be cuboidal or octagonal or spiky, so they have a different kind of shape. So these are waste crystals that the plant produces that often will help to deter predators from eating them. Give a yucky taste and can sometimes be toxic and sometimes actually hurt them. So we are going to, in class, we'll go through and name all the parts of the cells, and I can do a separate video on that too if you'd like to have a video just to look back at, but I think I will end there for the talk about different structures of eukaryotic cells primarily.